So I talked about last time, how do proteins get into the endomembrane system? And generally the process of feeding a protein backbone through these channels in the endoplasmic reticulum so that they can either fold up inside there completely, or that's also how we make proteins that are stuck into a membrane and having some segments on the cytosolic side, some segments on the other side. The topic for today is once we've gotten proteins either stitched into a membrane like this or put into the non-cytosolic compartment inside the ER, at that point they're in the endomembrane system. But they might need to travel to a distant part of the endomembrane system. And we talked about how some of the proteins in here are destined to be secreted outside the cell by exocytosis. So if the protein's final destination is outside the cell, it will enter the endomembrane system. If its final destination is a lysosome, it also will enter endomembrane system and sort of go out, 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 back, back, back. When we bud vesicles into the cell by endocytosis, they also enter this system. And so you can think of this as a separate but also continuous compartment. So it includes the endoplasmic reticulum, the Golgi apparatus, endosomes, which you may not have learned about before this class. I kind of think of this as like a holding cell or something. Like coming in, go to this building. If you're heading out, go to this building. Maybe like immigration or something. Some things coming into the cell country, some things leaving the cell country. And then vesicles are also considered part of that endomembrane system. There are a lot of little components of endomembrane systems, so getting from place to the correct location is quite a challenge for cells, and that's what we're going to look at today. Firstly, if we think about what a vesicle is, it's a little membrane-bound compartment of its own, and they're roughly spherical, sorta. If we put membrane lipids in solution, some of them will automatically form that vesicle like we talked about back in membrane dynamics. But notice that these vesicles are coming from existing compartments. They're budding off when they form, and then when they fuse, they merge with the existing membrane. So those two processes of budding off and merging are also important to understand how substances move. So here's a picture of a membrane, this line extending down through here. And it's a time-lapse picture of a vesicle actually budding off that membrane. And I like to think about this like the membrane is a balloon, and you could grab some of it and pinch it, as we say here, and you can make a little blob of it, but that requires some force to do that. And vesicles do not just magically bud off, they have to have force applied to them to make them bud off. And you can think of these proteins as doing that grabbing, pinching process. And we're going to call those molecules clathrin, adaptin, and dynamin. So in your mental word grouping, clathrin, adaptin, dynamin go with vesicles. And that also goes with the concept of exocytosis. Well, maybe not so much. But, so this is looking at vesicle formation in a cross section. This picture is looking at vesicle formation looking toward that process. So you can see here there are various stages of vesicles budding out. This one's almost finished. And what you're looking at is the caging or the arrangement of those grabbing proteins. So this surface with these proteins, these are clathrin that you're actually looking at. And this boring, smooth stuff in the background, that's the plasma membrane. So under here, there is plasma membrane also bulging out, but these protein partners are literally pulling the membrane out. And next we're going to look at that in a cartoon form. So here's sideways again. And this is depicting how this happens at the plasma membrane. So if we look where our labels are, this is extracellular, this is the plasma membrane at the cell boundary, and these vesicles are budding off to travel inward. So you might want to make a star or a note that this process, if we matched it up with this map, it would be right here. But there's not anything particularly different about getting a vesicle to form here and getting a vesicle to form here either. It's still pinching. It's still using the same components. This can just be done in different directions. So the whole point of making a vesicle is to send something in the cell. The cell will not just make vesicles randomly for no reason, because it takes energy to make them. So when they are formed, they will be carrying some kind of cargo molecules, which are shown by the red balls down here. And in this case, there is something floating outside the cell that the cell wants to bring inside for some reason. That's the cargo. I remember what she said about this being too loud on the video. Okay. So there's the cargo. And that cargo has to have a shape that binds to a specific receptor for the cargo. Otherwise, how can we make it get in the vesicle? So these blue molecules that are the receptors, these have really two jobs. They specifically bind cargo, and when they are bound to cargo, they're going to help assemble the vesicle cage. So I kind of think of these as a cage of proteins around the forming vesicle that are helping it form and pinching it. So these receptors, the cell has to have a lot of these receptors. Like there's not one receptor that can grab all the cargoes because that's a specific shape binding event. So there are a lot of receptors that are specific on one side for the cargo. If I drew a line through this and make that arrow come down, this is the part that specifically binds cargo. That makes sense. What does this part bind? Adaptin. Yeah. So this part binds adaptin. So if you were to look at all of these receptors and like compare them to each other, this region would be more conserved. This end would be less conserved. Because there's a large variety of cargoes, there are not a huge variety of adaptins. I might come back to that point later. So once the cargo is bound to the receptor other end is binding to this molecule called adaptin. And adaptin is like a linker. It's not interesting on its own. It's just a bridging protein. So it bridges between the specific receptor and this darker green molecule, which is clathrin. So all of these are proteins. Clathrin is forming the outer part of that vesicle cage that you see in this picture. So remember, this out here is clathrin, and now you know what would be under it. What's under it? Right under it. Adaptin. And then under that is a receptor protein that spans the membrane. It goes through the membrane. These receptor proteins would have had to be put into the membrane in the ER, like what we talked about last time.
So,
Yeah.
are cargo molecules just floating around outside the cell?
Yeah.
Okay.
The question is, are cargo molecules just floating around outside the cell? Pretty much. They would be in a pool of cargo molecules, whatever that is, out here. And I know this is being really vague about what those are, but it could be a huge variety of things. And a lot of times, this could be some kind of nutrient that the cell wants to gather from the outside. Yeah. So the receptors are going to, you know, when I talked about trying to pull a fabric and not rip it, you have to have something that spans both sides and provides a little bit of surface area on each side. The receptors are doing that too. So they're going to allow force to pull the membrane without ripping it, because the force is transmitted up through the receptor. So another thing that's going on here is that these are showing, in some aspect, cooperative binding. So notice, we've got several receptors binding the cargo. It's not just one. If we just had one, this wouldn't really work. So they're showing binding dynamics to where, like, neighbor binding is helping the whole thing assemble. And also, please note that it takes more than, what is that, 5, 6? A lot more than that to form a vesicle. A lot. But cooperative binding between cargo, receptor, adaptin, and clathrin will help form the vesicle. And where dynamin comes in, this one, the way I remember it is like dynamics is showing some kind of movement. Dynamin is what's really doing the pinching off process. Or the severing part of, all right, I've pulled this vesicle, I've got this membrane segment stretching out here. Dynamin is going to help sever that. And then once the vesicle is formed, it's formed. It's going to be fairly stable at that point. And it doesn't need clathrin and adaptin anymore, and they will go back and join the pools of clathrin and adaptin. And they could be reused to form new vesicles. So remember, these are just for the pinching. The receptor stays there. Dynamin is just to do the final pinch and sever the protein. Out of all of these three, clathrin is the one that is most prevalent on standardized tests. Clathrin goes with vesicles. Dynamin is involved, but another term that this book, I didn't see it used, was something called a clathrin-coated pit. I'm going to write that over here. Remember, that process could be happening here. All of that stuff, it could be happening here. And when they form on the exterior boundary of a cell and are going inward, those can be referred to as clathrin-coated pits. Because if you look from the outside, it's like they're sinking down. If we look from the other side. So if you come across that terminology, that's still talking about vesicle formation. At this point, we say that this is a naked transport vesicle, and it's got its cargo. The receptor is still bound there, and now the vesicle is floating away. This vesicle will not really float around in the cell very long. At this point, it's going to attach to motor proteins. And it's going to move through the cell along microtubules. So if you remember back to that excellent animation that we had, where there were these large blobby wavering vesicles, and there was a motor protein, and it was slowly like moving the vesicle along the microtubule, that's what really is going to happen to this vesicle next. It doesn't just float around for long. It's supposed to be going somewhere. It's too big to diffuse somewhere very quickly, so it's going to be using those motor proteins to move that way. And at the end of class, I'm going to show that segment of the video again, after we've gone through all this.
I have a question.
Mm-hmm.
So, do they have specific motor proteins that take them to their specific place?
When do we talk about this? We talk about motor proteins like in the next unit, but in short, the question was, are there specific motor proteins that take them to specific places? And the answer is not really, but now that we know that vesicles are moving on microtubules in between these places, there are motor proteins that take them outbound, and there are motor proteins that take them inbound. So there's general outward traffic, there's general inward traffic, but the motor proteins are just like semi-trucks or something that's not, they're going to take stuff in a direction.
But they don't have it.
That's not where the specificity comes from, yeah. But that's a good question. It would kind of be like, if I said that I want to go north, I'm going to get on 81 in the northbound lane. And then eventually, if I got close enough to where I'm going, I would be automatically attracted to that place. That's what we're going to talk about next. Like, as soon as I got close enough to DC, if I got sucked out of the car, that's kind of like, it's not making sense right now. Let me talk about this. So that's related to this next process. So we've gotten a vesicle made. It's got cargo. We pinched it. It's associated with a motor protein, and it's on a microtubule. That step is left out between here and here. But we come back to it later in the class because it's later in the book, honestly. And here it appears to be floating again. It's probably still associated with its motor protein, but it's gotten close to where it's going. It's gotten close now. And something else that is in this vesicle from the start, you might as well go back and draw it in on the previous one, is this red line depiction that's called a V snare. So now we're looking at a more interesting cargo. It's not just a red ball, it's got a real shape. The receptor is still laying semicircle. Getting a little closer, but this V snare would have been put in back here. It's just not shown. And what that is really the location tag for where the vesicle is supposed to go. The V snare. I myself don't know back here how the correct V snare gets in to the membrane. Yeah.
So it would just be on, it would automatically be on the vesicle?
It would be on the vesicle way early in its journey, let me just say.
Okay.
Yeah. And if the vesicle, every time the vesicle is leaving one compartment and going to another, it would have to get a new V-snare.
So
Does that make sense?
The snare makes the specificity for its.
You can think of the V-snare as making the specificity. Yeah. The V-snare is determining the location. Because if the cargo can't determine the location because there are multiple places that cargo could be needed. So this V-snare protein, it is spanning the membrane. It is stuck in the membrane. They are kind of weirdly extended outside the cell, and they actually flap around. They're probably alpha helical, if I had to guess. They flap around. So this is like waving around out here. And notice that the vesicle also has an extracellular protein on its surface called RAB. And so this, you can think of RAB as being kind of like Velcro. So RAB and the V-snare together are going to help this vesicle get from close to its location to literally fusing with its target membrane. So down here we're calling this the target membrane. And then the vesicle can unload its contents. So the target membrane could be the plasma membrane. It could be a cellular compartment membrane also. But we can think of this as the destination. If this cargo, this soluble cargo that was floating around, if it's supposed to go outside the cell and be secreted extracellularly, its target membrane would be the cytoplasmic membrane. That's where its destination is. So this system is depending on long, extended proteins at membranes. And the question is, can we get these close enough together to each other to get a vesicle to fuse with its target membrane? So there's one called RAB that's important in this, that interacts with a tethering protein. I mean, you can maybe think of this like octopus tentacles, or we don't really have a great human example of anything that's like this. Well, maybe if you go zip lining and they throw a rope out to you, they're trying to get you the last 10 feet. I watched my kids do this a couple weeks ago. You just got to get closer over here. You're almost here. You slow down too much. The tethering protein will interact with RAB. And these two are trying to get the vesicle close enough that the V snare, and on the V part, V stands for vesicle. And snare stands for something, but you will never see it spelled out unless you go into that research field. V stands for vesicle. And here's its V snare tag. And if these two can get close enough to the target membrane has a T snare that will specifically bind with the V snare. There they go. And they actually, they interact, and when they do, they also have a twisting motion. They wave, it's like they wave around, whoa, and then when they combine, they bind well, and then they start twisting. And the twisting pushes the vesicle close enough to the target membrane. If you can get the lipids to touch, they'll fuse. And once they fuse, and the cargo can come off.
So are V snare and T snare just found in the membrane?
You would not expect to find them anyplace else except membranes. Yeah. And they occur in pairs. So the T snare for a cytoplasmic membrane should only pair with a V snare that's supposed to go there. So if we imagine that a vesicle made it over here and it's got a V snare for a peroxisome, like it could get this close, but it's not gonna interact with this T snare because this is a cytoplasmic membrane T snare. That would just be tragic, wouldn't it? Got really close. This is the wrong membrane. This is the wrong building. I'm not even in the correct location. You would not have vesicle fusion. So you have to have matching of a matching V snare, a matching T snare to actually achieve vesicle fusion.
Do the carbon receptors just stay there, or do they get moved, or do they get reused?
They get recycled. Is that related to your paper? One time, somebody in seminar did a talk about cystic fibrosis that had something to do with that topic of what happens to the receptors. So, question is, the receptor, what happens to this now? And the receptors do get recycled. And the way that they do is pretty cool. The cell constantly brings in membrane from the outside as a default mechanism. And that has a couple of benefits. So there's constant low-level inbound trafficking of vesicles coming back toward the cell. Number one, to check what's out here. So when a vesicle forms and pinches, whatever is kind of in the liquid and happens to be floating there will also come in with it, even if it wasn't attached to a receptor. So that helps the cell check what's out here. It also helps the cell have a way to change out its membrane lipids. Like, they don't last forever. So having some of them come back into the cell and get degraded, that happens. But when leftover receptors are out here, they can eventually go back into the cell and be reused again through that inbound pathway. Oh, make a note here. Back on this slide again. This is a really good study slide, although I don't ask any specific questions about it, but it's good to be looking at when you think about these other processes. So the green arrows are sometimes referred to as the retrograde pathway or retrograde transport. And if you counted how many vesicles or how much vesicle traffic is going out with the red arrows versus how much vesicle traffic is coming in, the out is more prevalent. But there is a constant low level of retrograde transport in. And a lot of it is heading to lysosomes to digest materials, to help recycle them. But the early endosomes are also a little bit able to act like a decision maker. Say, oh, those receptors would be worth saving. Let's not trash them yet. But that's, if you hear of retrograde pathway, that's what that means, is the inbound traffic.
I have another question. I don't know how important it is, but when V snare and T snare, you said they become tightly bound.
Mm-hmm.
How would it become unbound?
Unbound?
To be used? Are there proteins that come into that?
Question is, once these become tightly bound, how do they get unbound?
Because they'd be used again, right?
They could be used again. Getting them, there would not be, I don't think, anything that I know of that would make them automatically come unbound once they fuse. So I'm not sure if these can be unraveled later If there is an unraveling protein, that may be possible. I'm not sure. But they would not just automatically become unbound because this just fused, because nothing would have changed in their environment. This, though, what made this come off? Why would the cargo now not be bound to the receptor when it has been all this time?
Diffusion.
It could have diffused into the vesicle too when it was in the vesicle. It is diffusion. I guess the question is, why is this diffusing away now when it didn't previously?
Is the receptor?
Are the conditions different between the two?
There's different conditions out here than there were in here. So that's when you go to biochem, you'll talk about how binding strength changes if the pH changes, if the salt concentration changes, if the positive, negative ionic balance changes. That's different out here. That's what makes the cargo come off. That's helpful. But the cell doesn't have to use energy to get the cargo to come off. It just does diffuse out once it reaches the extracellular conditions. Oh, here's another picture that would have been good to show earlier, but it's showing nicely that there is not just one V snare and one T snare. That would not be enough grabbing force anyway. There's multiple. If they can get close enough to interact, they do. When they do interact, they twist around each other, which forces the vesicle close enough to the membrane to get it to fuse. So, once it fuses, the cargo has been taken to the appropriate location. One other thing I want to say is that cells use this system to also just send new plasma membrane lipids out to the membrane. So we delivered cargo that was in the vesicle, but we also delivered new membrane lipids as well that went out here. So that process is kind of like changing water in a fish tank. Like you never take all the water out, so there's always some percentage of water in the tank that's technically old. But if you keep putting mostly new water, you can maintain the water overall. Instead of water, membrane lipids. Send some new, have it mix with the old, sometimes take back out old. Okay. That topic about V snares and T snares is really important for many kinds of processes in the cell. What I'm going to go through next are sort of some additional details of things that can happen within endomembrane system traffic. So remember that when we first made a protein that's supposed to be sent out to a location, let's think about cargo here. This is a protein. Doesn't have to be a protein, but we're going to assume it's a protein. If it's a protein, the cell made it. If the cell made it, it had a gene to encode it. I'm going to skip back several slides and back to here. Okay, so imagine now that this polypeptide chain that's coming out, that's the start of that cargo protein. And that protein, if it ended up in a vesicle, it had to go into the ER. Remember that? If it ended up in a vesicle, it had to get into a vesicle by being in the ER first. So it would have had this N-terminal signal tag. With that protein, the interesting cargo protein, would it have would it have one of these? Would it have a stop transfer sequence? It would not, because that protein was floating. It was soluble. It was not a membrane protein. So I want you to be able to connect this with what happens later and this with what happens before and not have them as isolated events, because they're not. So if we're making a soluble protein, a floating protein that will someday float in some compartment, we're making it back here, and then it has this long journey, maybe. Pretty long journey. We don't want to send it on the journey if it's bad to start with. This would be like a factory shipping bad product and paying to ship it when it's not even the right thing. So there is a quality control mechanism inside the ER to help check, are these proteins even folded correctly or not? So the inside of the ER lumen has several chaperone proteins in there, which are folding helpers. You can think of those as folding helpers and also shape checkers. Do these proteins have the correct shape? Before we're going to send this vesicle on a long, long journey. If in the ER, so here's a new picture that we haven't seen before. Here's the ER lumen. There's a ribosome translocating and translating, in this case, chaperone protein, because the chaperone itself had to get in there. It had to have a tag to signal it to get in there. If the cell is making, the cell can sense when it needs more chaperone proteins. Like if there's a lot of misfolding going on, the cell can make more chaperone proteins to help with that. And the way it does that is there are receptors basically for unstructured segments. Because if a protein isn't folding correctly, it's going to have a lot of unstructured segments, more than normal. The cell can sense that, and it can increase transcription of chaperone proteins. Like, if you work at the factory in Louisville, and your factory is shipping messed up mattress pads. This is where my uncle works. That's why I'm using this example. And his company did not ship messed up mattress pads. They're like really good. But if you were shipping messed up mattress pads, and word got back to the headquarters, you would send like more people, a team, to that location so that they can fix their problem. We need more chaperones. We're not folding correctly over here. That problem can be helped by having more chaperones. However, if the cell makes more chaperones, sends it to the ER, and that's still not enough, that's a signal that there is some kind of major problem going on. If we still have too much unfolded protein, the cell takes that as a signal that I am so messed up that I probably need to shut down. And the cell will undergo apoptosis. Book readers, what disease were they relating to this process? Insulin resistance, yes. So one place where this happens is in pancreatic beta cells if they're not making their insulin properly. Oh, you're not making your insulin properly? Let us send some more chaperones in there to help you with that. Oh, you still can't make insulin properly? You can detect that there's just too much chaos going on in here, and those cells need to be shut down. But they're supposed to be making insulin. Well, they weren't. Those cells would be shut down. And so a lot of this, the reason that we know this is because of diabetes research, getting funding to look into what's the problem. And sometimes the problem is this. I wonder if the people were surprised, like, ER, are you kidding me? That's what I would be like. That's the problem? There's not a problem with the insulin gene. If you looked at the insulin gene, it's okay. Probably. The problem is with folding.
So
Yeah.
this happens to a ton of cells for people who have diabetes.
I don't know if, I mean, it's happening to some of them. So there are certain particular cells in the pancreas that can make and secrete insulin routinely. So you have in your body a gene for making insulin in all your cells, but pancreatic beta cells are specialized to make a lot of it and secrete it quickly and massively. I think I might have some more slides about this. I'm going to skip ahead a little bit. So pancreatic beta cells are supposed to be making so much insulin that you can see it in a vesicle. That's amazing. So here's the cargo. There was actually so much insulin that it is not bound to the receptors. It's floating in the middle. Some of it would be bound to receptors, but there's just a lot more in the middle because of where it just came from. Like when this vesicle gathered, there was so much floating insulin, it wasn't attached to receptors. And they're supposed to be secreting it into the bloodstream, is where it's supposed to go. I feel like I got off track from your question. So if every cell has an insulin gene, pancreatic beta cells are set up to do more insulin production and secretion. And there are multiple of those. It's likely that if one cell has the problem, the others would too.
So is this with the ER not, like the chaperone protein's not working or not being sufficient? Are there other diseases besides like, diabetes that happen because of that?
There probably are.
Yes, there's,
Do you know other examples?
this is like a side step, but polycystic ovarian disease causes this. In a sense.
Causes this or results from this?
The hormones in your body, they kind of malfunction a bit.
Mm-hmm.
It causes insulin resistance
Mm-hmm.
and will lead to this.
Mm-hmm. I'm not sure if every cause of insulin resistance is related to this.
Just
And it's also likely that more diseases will be discovered to be related to this process. Where it would be most prevalent is if the cell is supposed to be making a lot of something and sending it through export pathways. Yes. So where this tends to go wrong is if you're supposed to make a whole lot of properly folded protein and export it, and that product is super important for the body, like insulin is, it makes sense that having a problem here would affect that a lot.
So this would be type 1, correct? Because type 2 is the receptors are an issue, right?
I'm not confident about that. I don't want to say more than I know. Okay, let's talk about next if the cell is making proteins that are headed for cytoplasmic membrane, and they're supposed to have a variety of sugar chains on them. So maybe let's put a little bit of information about properties of extracellular molecules. So if we are sending proteins that are going to either be secreted into the extracellular space or part of them sticking out into the extracellular space. They have a couple of different characteristics. Number one is that they often will encounter more, or let's say less controlled conditions. I mean, we hope that our extracellular tissue fluid is staying within balanced conditions, but it's harder to control conditions outside of a cell than it is inside of a cell. So this is like if we're gonna send astronauts outside the atmosphere, there's rougher conditions out there. Also, several of the proteins that go toward the extracellular space, many of them are going to need complicated sugar chains. And these have a few different purposes. One is that they hydrate the cell surface by sugar attracting water to the surface, and so they provide hydration and sliminess and slippability of cells to slip past each other. And the other is these sugar chains are used as identifiers. Like a human cell ought to have human type sugar chains on its surface. And like they can be really extensive and complex. So if a protein is supposed to have those, it's put on after it folds, not before. You can imagine, like if you've got this attached to your backbone and you're trying to fold, it would be in the way. It would be in the way. So after the protein is folded correctly, the ER lumen or other components of endomembrane system have enzymes that put the sugar chains on. This is useful because the lumen side of the membrane is going to eventually become the extracellular side. So that's where the enzymes are to put the sugar chains on, or other post-translational modifications. If we put sugar chains on, that's called glycosylation. Kind of sounds like glucose. Glycosylation to add sugar chains. And also, if the protein needs disulfide bonds, it's probably because it's going to a compartment that needs disulfide bonds.